Welcome everyone to the continuation of our innovative technologies segment. It is my pleasure to introduce Dr. Jing Xu, Assistant Professor of Obstetrics and Gynecology at the School of Medicine at Oregon National Primate Research Center and Center for Embryonic Cell and Gene Therapy. She earned her PhD degree in biochemistry and molecular biology and obtained her postdoctoral training in neuroscience and reproductive science at Oregon's Health and Science University. She was appointed a Birch Scholar, which is a very prestigious NICHD Building Interdisciplinary Research Careers in Women's Health Award, and became faculty member at Oregon Health and Science University in 2012. Dr. Xu's research investigates the production and direct action of paracrine factors and hormones in ovarian follicles using a non-human primate model, and I'm very excited to introduce her, or to welcome her to the podium. Thank you.
Thank you, Dr. Loranda, for the introduction. Good morning, and I want to thank the Oncofertility Consortium for giving me this opportunity to share with you the research progress in vitro maturation of primordial follicles to yield eggs. As introduced by Dr. Woodruff, in vitro follicle maturation is to provide options for fertility preservation in patients with POI. POI is characterized by the reduced ovarian reserve or the decreased number of follicles in the ovary. Follicles are basic functional units in the ovary. Each follicle contains, it's really hard. No, I don't think I have a pointer here. That's okay. Each follicle contains an oocyte surrounded by follicular cells. While the oocyte is responsible for fertility, follicular cells produce ovarian steroids to support endocrine function. So pediatric patients with POI may experience delayed puberty, and adult patients may experience early menopause and infertility. And the possible cause for POI include genetic disorders such as Turner syndrome and sickle cell disease, autoimmune disease, and ovarian toxic medical treatment as introduced by Dr. Kim, and exposure to hazardous substances. Current fertility preservation options for patients with POI include embryo, egg, and ovarian tissue cryopreservation. And ovarian tissue cryopreservation is more practical for pediatric patients and patients requiring immediate ovarian toxic medical treatment. Ovarian tissue cryopreservation is to preserve primordial follicles located in the ovarian cortex. As shown in this picture, the primordial follicles are on the lower bottom to the left. A primordial follicle contains a primordial oocyte surrounded by a single layer of flat pregranular cells. When primordial follicles are activated, pregranular cells become cuboidal granular cells. During further granular cell proliferation, there will be multiple layers of granular cells surrounding the follicle, and the follicle become from primary to secondary and to multi-layer preantral follicles. And then an antral cavity is formed in the granular cell layer. The follicle become antral follicles. Then the granular cells differentiate to form cumulus cells surrounding the oocyte, and mural granular cells lining the follicle wall. In women, each cycle, one of the antral follicles is selected for continued growth and maturation to produce mature oocytes for fertilization. So in order for the primordial follicles to generate mature oocyte or egg, one possibility is to transplant the ovarian tissue back to the patient. As mentioned by Dr. Woodruff, there is invasive procedure, and there's a risk of reintroducing malignant cells back to the patients. That's why in vitro follicle maturation is developed to support follicular development in vitro to yield mature oocytes or eggs. In vitro follicle maturation is successful in mice using a multi-step protocol. So first step, ovaries from fetal ovaries are cultured as a whole organ. Primordial follicles are activated and grow to the pre-antral stage in 8 days. Then oocyte granulosa cell complexes are isolated from the cultured organ for another 8 days of culture. And as you can see in the first picture on the bottom, healthy germinal vascular GV oocytes were harvested from the culture. And when the oocytes underwent in vitro maturation, or IVM, with exposure to gonadotropin, the oocytes matured to metaphase 2 or M2 stage in 17 days. And then after in vitro fertilization, those oocytes were fertilized and divided and support the further embryonic development to early embryonic stage. After embryo transferred, live offspring, a total of 60 live offspring were obtained by these two studies. To date, 2 research groups reported in vitro follicle maturation from primordial follicles using human ovarian tissue. Different from the work in mice, this multi-step protocol starts with cortical tissue culture. And the primordial follicles were activated and grow to the pre-antral stage. And then pre-antral follicles were isolated for additional individual follicle culture and to the antral stage. And then cumulus oocyte complexes were isolated for IVM. Metaphase 2 oocytes were obtained, although if you look at the metaphase 2 oocyte picture, although the polar body appeared to be larger, relatively large compared to the metaphase 2 oocyte, the spindle structure was typical in this case. And another study was reported by our research group using the similar 3-step culture protocol. The only difference is the group culture is used for the second step. And I'll get into the details. The first step is to activate primordial follicles in the ovarian tissue. Primordial follicles can be activated spontaneously in vitro. Although ovarian tissue or the ovary were cultured in serum-free medium, a portion of primordial follicles are activated in cultured mouse ovary. And all primordial follicles were activated in cultured ovarian cortex from cows and monkeys. The reason for that could be the removal of inhibitory signals, such as the Hippo-YEP signaling pathway. And another is, as mentioned by Dr. Hayashi, and the alteration of a physical hypoxia state of the follicles. As you can see in this picture, the top one is from the mouse ovary, the bottom is from monkey ovary. The expression of carbonate anhydrase 9, which is a intracellular indicator of hypoxia, the brown color indicating positive signaling. CA9 expression are located in the primordial oocyte and granular cells in both the mouse ovary and the monkey ovary. And when we do ovarian organ culture or ovarian tissue culture, those organ or tissue are cultured at 20% oxygen. But the in vivo, the ovarian cortex is avascular region. Oocyte concentration is around 1 to 2%. So the increased primordial follicle activation during ovarian tissue culture could be due to the high oxygen. And for the monkeys and the cows, the increased primordial follicle activation could be due to the oxygenation during tissue processing. Another question is, what's the marker for primordial follicle activation? I want to introduce anti-Mullerian hormone, AMH. AMH is produced by granular cells in growing follicles. As you can see in this orange section from a monkey ovary, PM stands for primordial follicles. Pre-granular cells do not produce AMH. AMH start to become evident when pre-granular cells become cuboidal granular cells in primary follicles. When the preantral follicles grow larger, they produce higher levels of AMH, and when after antral formation, AMH expression decreases. So AMH is detectable in the culture media when there's only one single preantral primary follicles are cultured in the culture dish. So AMH is very sensitive and non-invasive marker for primordial follicle activation. This picture is to show you a human primordial follicle, which is tightly associated with stroma. To activate primordial follicles, cortical tissue was cut into small fragments with a diameter around 300 micron, each piece containing more than 5 primordial follicles. In 3 weeks, primordial follicles grow to the preantral stage. As you can see, the preantral follicle in this section contains a healthy GV oocytes with multiple layers of granular cells and intact basement membrane. The enlarged picture is to show you that the physical support from the stroma is critical for early growth of the follicles. As you can see, at the end of the week 1 of culture, the follicle located in the middle of the tissue has an intact single layer of granular cells. However, the one close to the edge, granular cells migrate away from the oocyte. AMH production was not detectable in the first week of culture, and the concentration increased in week 2 and plateaued in week 3, which means primordial follicles stayed quiescent during the first week of culture. They're activated in the second week and the primordial follicles become preantral follicles, and they grow larger to produce increased level of AMH. And then by the time of the 3rd week, it's time to isolate those follicles for continued follicle culture. And indeed, AMH expression was detectable in the developing preantral follicle, as shown in the brown color. Next step is to culture primordial follicles to the antral stage. There are several important factors during this stage. First one is gonadotropin, such as FSH. Although preantral follicle growth is thought to be FSH independent in vivo, FSH is required for human preantral follicle survival in vitro. A naturally occurring FSH is a mixture of FSH forms, different forms. Although those FSH forms have the same peptide structure, they have different glycosylation patterns due to post-translational modifications. Currently, there are 3 types of FSH are used in the FF clinic. The first one is highly purified urinary-derived FSH. And in the US, human recombinant FSH alpha and beta are used for ovarian stimulation. Compared to natural FSH, recombinant FSH have shorter sugar chains. And FSH alpha is more acidic than FSH beta. So when monkey follicles were cultured in dish with the 3 different kind of FSH, the natural FSH group gave us the highest antral follicle formation rate. So FSH dose need to be adjusted depending on the type that is used. Another factor is paracrine factor regulation need to be considered. I give an example of AMH. Our study showed that AMH promotes preantral follicle growth and inhibit antral follicle maturation. As you can see in the top panel, the figure in the middle, Cultured monkey follicles usually form an antrum from the pre-antral stage to the antral stage, from the antrum at culture week 3. With addition of MH, antral formation started early in week 2. The bottom panel showed estradiol production by the cultured follicles after antral formation. After antral formation, the control cultured follicles produce high amount of estradiol. After MH addition, estradiol production is inhibited. So from there, we can modulate MH in the culture media, MH addition during the pre-antral stage, and depletion during the antral stage may promote follicle growth in vitro. There are several markers can be used to assess follicle growth in vitro. For pre-antral follicles, MH is a great marker, as shown in this figure. The top line is the growing, refers to growing follicles. MH production increases during pre-antral follicle growth. After antral formation, MH concentration in the media plateaued and then decreased once the antral follicle grow larger. This production pattern is similar to what happens in vivo developed follicles. The bottom line is no growing follicles. If the follicle don't grow, they don't form antrum, MH production remain the same over the 5 weeks of individual follicle culture. For antral follicles, estradiol production can be used as a marker, as you can see in the figure on the right. Estradiol level increased after antral formation, and the larger follicles produce higher levels of estradiol. In addition, actin A, I didn't mention inhibin B follow the same production pattern as AMH for pre-antral follicles, and activin A, IGF-2, VEGF follow the same expression pattern as the estradiol. So the follicle, pre-antral follicles can be cultured individually to the antral stage in 6 weeks. That's human follicles. The survival rate of follicle is about 65%. Follicles can die in the middle of the culture, either during the pre-antral stage or during the antral stage. Follicle death can start with oocyte degeneration, as shown on the top panel, or granular cell apoptosis, as shown in the bottom panels, to extrude the oocyte. For surviving follicles, the isolated pre-antral follicles can grow to the small antral stage with increase that for about 4.5 increase in diameter. And when you look at the MH production in the media, MH production increased during pre-antral follicle growth, plateaued after antral formation, and decreased in large antral follicles. And the histology staining showed an oocyte, healthy oocyte surrounded by cumular cells and multiple layers of granular cells. Pre-antral follicles can also be cultured in groups of 3 to 5. Those isolated follicles form aggregated together and form an organoid-like structure during the first week of culture. By the time of the 6th week of culture, those follicles form antrum to become antral follicles, as shown in the picture. As you can see in the histology staining, there's a healthy GV oocyte surrounded by cumular cells with multiple layers of granular cells, mirror granular cells, and antral space. There are 2 antral follicles there. The enlarged picture shows, I want to show you, this is a cell, a cumular cell at metaphase stage of mitosis. The actively dividing cells are pretty common in mouse ovarinsections, but it's very rare in human ovarinsections. So this indicating that cells are actively dividing in vitro during in vitro development. And also there are aromatase encoded by CYP19 expression in granular cells, which are responsible for estrogen production. and 7-alpha hydroxylase encoded by CYP17 expression in thecal cells, the brown color in the picture, which are responsible for androgen production. Finally, oocyte maturation. In order for the germinal-vesicle oocyte to mature to the metaphase to M2 stage, one strategy is to apply gonadotropin stimulation to cultured antral follicles. As you can see in the picture, that's the monkey follicles. If cultured monkey follicles, the antral follicles have a well-developed cumulus oocyte complex, or COC, gonadotropin stimulation can trigger ovulation in the culture dish to produce metaphase 2 oocyte. Another strategy is to do IVM, that is cumulus oocyte complex culture. Because we cannot fertilize human oocytes, the oocyte quality assessment can be done by polarized light microscopy, which is an invasive procedure to look at the meiotic spindle position, immunofluorescence staining to look at the meiotic spindle morphology, and transmission electron microscopy to assess M2 oocyte-specific ultrastructures, such as microvilli, corticosgranules, and mitochondrial distribution. In addition, epigenetic and gene expressions can be analyzed as well. To date, we have not been able to obtain M2 oocytes from individually cultured human follicles. Although the cultured antral follicles have well-developed COCs, after 34 hours of HCG and EGF treatment, all oocytes remain at the GV stage. You can see that in the immunofluorescence staining, the GV nucleus, there were abundant transrenal projections showing the connection between cumulus cells and oocyte. The reason for them not achieving meiotic maturation could be that the sizes are relatively small. In this case, 105 micron in average, which is relatively small in the human oocyte. But we were able to obtain metaphase 2 oocyte from group cultured follicles. After we did IVM for 48 hours, and some of the oocytes had, the COCs had cumulus cell expansion, but the oocytes remain at a GV stage. Some of them don't respond, they still have compact cumulus cells. But about 20% mature to metaphase 2 stage. As you can see, it has, the oocytes have proper size, polar body size and meiotic spindle position as shown in the red picture, the post-scope picture. And electron microscopy show the chromosome alignment in the meiotic spindle, cortical granules under the plasma membrane of the oocyte, and the microvilli projection out of the on the oocyte basement membrane. There are round and elongated mitochondria associated with vesicles and smooth ER, which are typical for M2 oocytes. To summarize, in vitro follicle maturation supports human follicular development from the primordial to the antral stage to produce oocytes competent for meiotic maturation. Currently, multi-step protocol is needed, including ovarian cortical tissue culture, three-dimensional follicle culture, and oocyte in vitro maturation. In vitro follicle maturation protocol can be further optimized to improve follicular development and oocyte competence. For example, we haven't provided any stimulatory factors to the primordial follicles yet. So stimulatory factors can be applied to promote primordial follicle activation. And also, in addition to MH, other paracrine factors or regulatory factors can be considered to promote pre-antral to antral follicle growth in the second step. Omics studies such as genomics, proteomics, metabolomics will be helpful to identify critical factors during this follicle growth stage. And oocyte quality can be further analyzed by comparing in vitro and in vivo developed oocytes. And of course, for clinical applications, in vitro follicle maturation need to be done using cryopreserved and recovered ovarian tissue. Using the protocol developed by Dr. Mary Zielinski, we're able to cryopreserve human ovarian tissue. You can see the typical morphology of primordial follicles in cryopreserved recovered ovarian tissue. And after 1 week of tissue culture, primordial follicles were activated as shown in BRDU staining, which labeled actively dividing cells. As you can see, some of the pre-granular cells become granular cells. After 3 weeks of culture, we can see multi-layer pre-antral follicles develop in those little cortical tissues. Of course, next step will be isolating those pre-antral follicles for subsequent follicle, in vitro follicle growth. I would like to thank my previous lab staff, trainees, and collaborators at Oregon Health Science University, and the technical support from the research course and my previous colleagues at OHSU. And the work was done with financial support from NIH. Thank you for your attention. I'm happy to take your questions.
This was a great talk. You did answer part of my question at the very end in your summary, but I had a question about the cortical culture. You said that you don't add factors to that culture at this point, but is there serum in the medium?
There's serum-free medium, yes.
Okay, so basically just base medium and nothing else added.
Base medium with FSH. So with FSH, insulin, selenium, yeah, the basic stuff.
So that's the condition for the development from primordial to primary follicle in the cortical culture.
Right, but I don't think FSH at that point is critical. We put FSH because once the primordial follicles are activated, they become preantral follicles. We want to support preantral follicle survival. Mm-hmm. That's why we put FSH, but I don't think FSH is critical for primordial follicle activation.
All right, thanks.
Yeah, thank you.
Thank you for very nice talk. So I'm just have one question. So what happens if you puncture the antral follicles artificially? What happens in the oocyte and the COC in the human?
Culture COC, you mean?
Yeah.
If
you puncture you culture the 3D culture so that you can keep the structure of the follicles. Right. But what happens if you break the structure?
Follicles?
Yeah. What's happening in the oocyte?
So the early stage follicles, like pre-antral follicles from humans, if you culture them on the traditional, let's say, conventional culture dish, granule cells migrate from away from the oocyte, and the oocyte will be exposed, and then the follicle will not survive. The oocyte die as well. So that's why the pre-antral follicles need to be cultured in 3D to maintain the 3D architecture. We use low attachment plates, so you don't need matrix embedding at all. So they just keep their 3D architecture. After pre-antral follicles develop to antral stage, you can isolate the COCs out of the antral follicle and culture them in just the common culture dish. It can survive. The COC culture can survive, 2D culture.
But what is the percentage of the becoming the M2 oocyte? So that's better in the...
become M2s if the COCs were isolated from in vivo developed small antral follicles, similar size. The maturation rate can be 90% in our condition.
Thank you.
But for in vitro Currently, as you can see, around 20%,
Okay.
similar size. That's, I will say, between 1 to 2 millimeter size.
Thank you.
Thank you.